Welcome, everybody. My name is Mike Nickerson. I am a professor in the Department of Food and Bioproduct Sciences. This is our second installment of the Capture Seminar Series, which is a research and training collaboration between the University of Saskatchewan, Manitoba, and Alberta. So today we're really excited to have Dr. Supratim Ghosh give his seminar or webinar on plant protein stabilized nanoemulsions and structuring them into foods. Dr. Ghosh is an associate professor within the Department of Food and Bioproduct Sciences at the University of Saskatchewan. He received his PhD from Pennsylvania University in 2007 and did his postdoctoral research at Ryerson University. His research interest is in emulsion and colloidal science, including food chemistry, structure-function relationships, food nanotechnology, lipid oxidation and emulsions, and complex colloidal chemistry. His team has been working on utilizing plant proteins to create structured food emulsions, oleogels, to deliver bioactive via nanoemulsions for health benefits and sustainability in food production. I'd ask everyone to write questions in the chat or save them at the end, and maybe keep your cameras off and your mics off during the talk. Thank you, Dr. Ghosh, you can take it away.
Thank you, Dr. Nickerson, and thanks for your kind introduction, and thanks for your invitation to give this talk. It's really exciting. So let me share. So I will start with this. My daughter took this photo of different pulses that we have at our home, and she made sure that I shared this, and I mentioned give credit to her for this photo. So, okay, I thought it will work, and so today's talk is on utilization of plant proteins for structuring emulsion-based food. And so we will talk about a brief overview of emulsion structure, stability, and their food application. And then a little bit more on how you can create structure in emulsion, how you can transform a liquid emulsion into an elastic gel. We will talk about different types of emulsion gels, and then we will talk about how you can use controlled aggregation of protein stabilized emulsion droplets to create structure in emulsion. So after this brief introduction, we will talk about 3 examples from our lab. First one we will talk is on canola protein stabilized, highly concentrated emulsion for oil structuring. Then we will talk about another research where we use faba bean protein to create emulsion gel to replace animal fat in meat. And last, we'll talk about some latest research where we were able to create pea protein nanoparticle and use them to improve emulsion stability. So to start with emulsions, as we all know that these are dispersion of 2 immiscible liquids, oil and water, where one phase is dispersed as small spherical droplets in the second. So oil in water emulsion, so oil droplets are the dispersed phase and dispersed in the continuous phase. And many food in our daily life, milk, mayonnaise, coffee creamer, flavored beverages, salad dressing, and you name it, mostly they are made of oil in water emulsion. There is another kind of emulsion, just the opposite of that, water droplets dispersed in oil phase. And the 2 most prominent example in our food are butter and margarine. So they both are oil in water in oil, rather water in fat, where the oil phase is a mixture of fat crystals and liquid and solid fat. Now, how emulsion droplets are stabilized? Again, I'm sure all of you know about this, but still, let's have a look. So if we are able to look at the interface, mostly that oil droplets are stabilized with amphiphilic molecule called emulsifier, and they have 2 prominent part. One is the hydrophilic part, which is water-loving part. It like to stay in the water phase. and the hydrophobic part, that oil-loving part that like to stay in the oil phase. So this is like a standard, emulsifier. Mostly we call them small molecule emulsifier. So some of the prominent examples from food grade emulsifier that are used in our daily food supplies: lecithin, where you have the hydrophobic part of the triglyceride part, and then you have the phosphatidylcholine part, the phosphate group. So that's the hydrophilic part. Twin 20 is a synthetic emulsifier that been used for a long time in food. Nowadays, with a lot of concern about using synthetic emulsifier, probably they are used much less. So it still also have a hydrophilic part and then hydrophobic part, the fatty acid chain. And monoglyceride, they are oil-soluble emulsifiers, so they are mostly used for margarine manufacture. So they similarly they have a hydrophilic part, the triglycerol part, and then the fatty acid part and the hydrophobic part. So these are small molecule emulsifiers. But we are in this talk today mostly interested in protein, right? So proteins are, we can call them polymeric emulsifier. So most commonly, we are all aware of milk proteins, whey protein, casein protein. They have been in our food supplies as a food ingredient for a long, long time. Egg protein is another very commonly used protein in our food. Lecithin and egg lecithin. Mayonnaise are stabilized with egg protein and egg phospholipids. But nowadays, obviously, a major food ingredient, emerging ingredient, if you can call it, plant proteins. So, some, lentil, pea, various beans, so you can extract the protein. So now, the different proteins, they can unfold to different extent at the oil droplet surface, and that leads to different kind of stability. If you can, like a very common, like traditional emulsifier, sodium caseinate, if you can, it is commonly believed that if they are at the interface, they are disordered state. They take a position like a train, loop, and tail positions. So where the part would be in the oil phase, hydrophobic amino acid portion, and hydrophilic amino acid portion would be outside in the aqueous phase. Now, globular protein probably are more relevant for our plant protein-based emulsion. They are different shape, right? They are more compact structure, but it is commonly believed that they also keep, they little bit denature, open their structure when they absorbed on the oil droplet surface, but still they can keep somewhat their globular shape. And because of that, also they can create a dense structure at the interface. So, whey protein, pea protein, most plant proteins have this kind of globular protein structure. So with that background, let's look into a little bit before we move into the emulsion structure, emulsion destabilization mechanisms. So, and this will help us understand some of the things that we are going to talk later. So if you have a kinetically stable oil in water emulsion that are stable for a reasonable amount of time, but because this oil droplet, they are lighter than water, due to gravity, the oil droplet moves to the top. So we call it creaming. Now, if you don't have creaming, but the oil droplet can also feel attraction between them, due to various reasons that we will talk in next few slides, but droplets can aggregate. Now, the challenge is to prevent at this step. If you can stop it at the droplet flocculation, that's great. And we will talk that how useful this is. But the coalescence, where the droplet fuse together, that's the ultimate destabilization mechanism. And we don't want to have that if you want to have a stable emulsion in our food. In the creaming also, when the oil droplet cream, they are more close to each other. There is a greater chance of coalescence, and eventually that can lead to oil separation on the top of your food emulsion. That definitely is the worst thing that can happen. You don't want that. So now, traditionally for beverage emulsion, for liquid emulsion, we don't want creaming, we don't want flocculation as well. But when you are talking about structure emulsion, things are a little more interesting, and let's see how it turns out. So in the structured emulsion, then they are semi-solid. That's why we are calling them structured emulsion. And it does not flow on its own, can behave like a solid until a high shear is applied. A great example would be, mayonnaise, that solid-like material, but when you take it on a knife and apply on a piece of bread, you can easily do that. So this kind of structured emulsion, also we can call them emulsion gels. Now, if structurally, there are three types of emulsion gel. The first one would be emulsion filled protein gel. So where there is a protein in the continuous phase oil that is also forming a network. Maybe I will turn on the highlighter. That is also forming a network, and then the oil droplet in between the protein gel, they either help or either does not help in gelation. So sometimes the oil droplet take part actively in the gelation, so we call them active filler, or they can be inactive filler also. So, emulsified processed meat could be a great example of this kind of emulsion filled protein gel. Yogurt is another example. Um, you can also get emulsion gel even if you don't have any continuous phase protein. You can have a strong attraction between the droplet, and the droplet will be attracted to each other and form a three-dimensional network, and can also hold water in between them and give you a gel structure. So you can call it emulsion stabilized protein gel. And Professor Dickinson has been working on this for a long time, and some of these concepts taken from his paper, a review paper published in Food Hydrocolloids. And the last one is our gelation due to close packing of droplets. So the best example here is mayonnaise, where you can just pack and pack and pack oil droplet until they touch their surface, and you can no longer pack anymore. And so they create a close pack structure. And because of that, they become a solid-like material, gel material. So these three kinds of emulsion gel, and today talk a little bit about each of them. Now, as I said that, to create the gel structure, that the droplets needs to feel some aggregation and attraction. So droplet aggregation is key to structuring in emulsion. Now, the kinetic stability of an emulsion is governed by their interdroplet interaction. So the overall interaction between the droplet, either attractive or repulsive, you can describe by various colloidal and non-equilibrium forces acting among the droplet and with the continuous phase constituents. For example, you can have, there is always attractive van der Waals flows, there can be attractive depletion interactions, and there is always hydrophobic interaction between the oil droplet. You can always also have repulsive electrostatic interactions where the droplets have charge and they repulse each other. Or also you can have steric repulsion where you have a very large biopolymer at the oil droplet surface. They prevent another droplet from coming close to each other. So these are the interdroplet interactions that you can control to create structured emulsion. And these forces depends on what is the emulsifier adsorbed on the surface of the droplet, and the continuous phase compositions, and also droplet size and droplet charge. But these are all interrelated. What kind of emulsifier adsorbed on the droplet surface, that will dictate droplet charge, and what kind of composition of the continuous phase, that dictates also droplet attraction, droplet size. So they are all interrelated factor. Now, let's talk a little bit on effect of interfacial protein on droplet aggregation. So we are focusing now on only on protein stabilized droplet. Now, any factor which tend to increase protein association in solution will cause protein stabilized emulsion to flocculate. For example, we know that if you heat, protein can denature, and then they open up their structure, and then they can aggregate upon cooling. Salt can also screen charge, can lead to aggregation. Enzyme in cheese making, enzyme also they induce protein aggregation, And pH in yogurt, the pH drop to isoelectric point and then lead to aggregation of protein. Now, all these factors that leads to this kind of protein aggregation, if if the proteins are present on oil droplet surface, and if you induce this kind of factor that influence protein association, the droplet will also aggregate. And this is the basics of forming structure in emulsion. So, so if you have a stable emulsion, you induce some attractive interactions and the droplet become flocculated. Now, this flocculated droplet, they behave like a large droplet, large entity, and they move on their own. And because of that, the viscosity of the emulsion increased tremendously. So just by inducing attraction between the oil droplet, you can convert a liquid emulsion into a very viscous emulsion. Now, there is a challenge also, though. This because they are now large entities, they can easily cream. They can go to the top easily, and you have a phase separation. So you don't want that. So now this kind of aggregation can happen due to weak attraction because droplets are not strongly attracted. They have some weak attraction. So this can lead to this kind of phase separation. On the other hand, if you have a strong attraction among the droplet, now here the droplet network are self-supporting and they can span the entire container, entire area, and that can hold water and then give you the gel-like structure. So here, when the droplet touch each other and they just stick because they have a strong aggregation, they cannot move around. So the structure is like really tenuous network, open network structure. So this is the key to create, either you how to create strong attraction between the droplet and create the gel structure. Now, we have to keep in mind the ultimate destabilization, coalescence, right? Because we are talking about that we are going to induce droplet attraction. That means droplet will be touching each other, and that can lead to the rupture of the interdroplet film and lead to coalescence. So that we have to prevent if we want to create structured emulsion in this. So that's why, for a structured emulsion with aggregated droplet, it is absolutely critical to prevent coalescence. So we have to make sure that the coalescence is prevented. Now, how can you control then? Have droplet aggregation but not coalescence? You can decrease droplet size. Smaller droplets are more stable to coalescence than larger droplet. You can control droplet movement. You can increase viscosity of the continuous phase so that you can prevent creaming and undesirable phase separation. But ultimately, you need to make sure that you have to control the film rupture to prevent droplet fusion. So, but you can increase emulsifier surface absorption and charge. So more emulsifier gets absorbed. So you add more emulsive protein to make the emulsion. But it's not that if you add more, you always get more stable. There are obviously challenging other factors. But absolutely, you need to make sure that the thickness of the interfacial layer is enough. So, for example, protein or biopolymer coated droplets are more stable to interfacial film rupture. And that's why, protein are great candidate to create this kind of structured emulsion. So, and pulse protein are obviously really good in this case. So with that, so I'm going to move into focus into some research data from our lab. We'll talk about three areas, as I said. Canola protein stabilized highly concentrated emulsion and fababin protein stabilized emulsion gel that we use to replace animal fat in meat. And if we have time, we will also talk about protein nanoparticle that can actually improve emulsion stability. So let's talk first about some of the highly concentrated emulsion structure. Mayonnaise is one of the example, as I said. It has about 66% oil in water. So it's a highly concentrated emulsion, as I said before. Now, droplets in mayonnaise are tightly packed together, and that gives you that gel-like structure. Now, we have to make sure then that the interfacial stability is absolutely critical. So the interface of the droplet must be strong enough to give you a stable emulsion. So the question then we ask, can plant protein be used to create similar emulsion structure with concentrated oil droplet? And because you're talking about how droplet concentration can increase emulsion gelation, so here is a slide that if you have a less droplet, it's a dilute emulsion. As you increase oil concentration, they become packed, and around 64% droplet. If the droplets are same size, monodisperse, their surface touch each other, and you reach a gel state. And if you still add more, you get a more compressed droplet structure. So based on the droplet concentration, you can convert a dilute emulsion into a viscous salad dressing, viscoelastic product, and then ultimately an elastic material like mayonnaise you can create. Now, in our research, this canola protein research done by Yan Ren Tang, who just defended her master's thesis on this work as of last Friday. So congratulations, Yan Ren, if you are listening. So what Yan Ren did, she extracted canola protein isolate from cold pressed canola meal using a salt extraction process. We use cold press because the proteins were really good. They were not denatured due to heat processing of the regular canola meal. And then the CPI was used to make concentrated emulsion. So we were able to make 50% canola oil in water emulsion, and we use 4 different protein concentration and at pH 7. And then we used a high pressure homogenizer to make the emulsion. Then after making the emulsion, as I said, that we need to introduce some factor that can induce droplet attraction. So we did that by 4 different ways. We added salt, we added vinegar. Vinegar was made by, as a 5% acetic acid. We wanted to create a composition somewhat similar to a salad dressing type of structure. And then we added both, because in food, you always have both. So we wanted to see effect of both salt and vinegar. And then separately, we also did heat treatment and to see how that can influence emulsion. This research has already been published in Food Hydrocolloids. Just wanted to mention that. And then we characterize the emulsion using various methodologies that I'm going to discuss. So straight away, I'm just showing you data for 1 and 4% canola protein stabilized emulsion. The 1% emulsion was viscous because it has 50% oil, but still flowable. So it can flow. However, the 4% CPI emulsion was really strong gel. Now why? And we will look at some of the data later. When you added salt, salt didn't do much. Salt definitely induced some attraction, but didn't change much of the visual observation. When we added vinegar, the acid pH was 3.7. You can see some emulsion destabilization. So the aqueous phase separated, probably some extensive creaming happened in this emulsion. Now, that is an interesting observation that with 4% CPI, the solid emulsion, they become liquid, viscous, like much more liquid than the 1% fresh emulsion. And then we added both. And then we saw that when you added both salt and vinegar, even the 1% fresh emulsion become a relatively strong gel, and the 4% also become very strong gel. So now from a liquid, then you can add this regular food ingredient to create a structured and elastic emulsion. So if you look at the structure of the emulsion, then this is a confocal microscopy. So we at 1%, the emulsion was not very stable, and we could not see much interesting effect. At 4%, though, one thing you can clearly notice the droplet size. The droplet was much smaller with 4% protein, and that created a better structure and gave us more stable emulsion and increased the viscosity or viscoelasticity. And then when you added salt, there are some attraction between the droplet, but still they can flow. Vinegar made the droplet, more not attracted to each other, not very clear from the microscopy. Heating, though, you can see that the green here are the protein and the red are oil droplet. Extensive aggregation between the droplet and the protein also covering the droplet. So, no, sorry, this is not heating. By adding salt and vinegar, you see more aggregation between the droplet. So we measure droplet size. So this is the average droplet size of fresh emulsion. So different protein concentration on the x-axis and different treatment. So we saw that emulsions, as you increase protein, their droplet size decrease. When you add salt or vinegar, not much change, but when you have both, there is an increase in average droplet size. Now, this was measured using a static laser diffraction droplet size analyzer. So you have a large droplet that could also mean that they are aggregated. So then we added sodium dodecyl sulfate to break up the aggregation, and you see that the droplet size is actually constant no matter what treatment you do. So that told us that so the droplets were very stable. So no coalescence happened even when you add both. But because of addition of salt and vinegar, the droplet size aggregation significantly increased. And that aggregated structure gave us the gel-like emulsion. So we looked at droplet charge. So initially, the fresh emulsion has a negative charge around -10 as a function of not much change with increase in CPI. When you added salt, charge definitely decreased. But then when we added vinegar, the charge reversed. Now it is the other side of the isoelectric point. Uh, so it should be pH 3.7, not 7. But the charge was very high. And because the charge was higher, the droplet can repulse each other and they were flowing easily because they are no longer attracted to each other. When we added both salt and vinegar, the acidic pH introduced protein denaturation, protein hydrophobicity increased under acidic pH. And because you have salt, the charge also decreased, and that created an really ideal scenario to create structured emulsion. We also looked at the creaming velocity. So I'm going to skip this just for the time sake, so we can measure the creaming rate using this. And we found that the creaming rate also decreased as you increased canola protein concentration, but the aggregated emulsion under this accelerated gravitation, they creamed a little higher rate compared to the fresh emulsion. We then focused into viscoelasticity. So these are the viscoelasticity data for as a function of strain. And then we took the data under the linear viscoelastic region at 0.1% strain and plotted them. Here's the interesting observation was that let's look at the 4%, the focus here. So fresh emulsion was highly elastic material. As you added salt and vinegar, their gel strength actually decreased. But when you have both, that's when the emulsion become highly elastic and strong gel and structured emulsion, and their gel strength is highest when you have both vinegar and salt. Then we heated those emulsions, and heating introduced a more interesting structure. So you can see that the 4%, this is the unheated emulsion. As soon as you heat, they are G prime. This is the G prime. The storage modulus increase significantly, and their crossover strain happen at a higher strain. So the gel become very strong after heating. So what happened is when you have a emulsion, concentrated emulsion structure stabilized with this canola protein, uh, the protein denatured due to heating. And because the droplets are covered with protein, then they are strongly aggregated. So now you can see the droplets are strongly aggregated, forming a 3D network, and the protein also supporting the droplet aggregation. So that created a really strong structure like tofu. Yanran tells me that this structure of 4% CPI heated is become a tofu-like strong structure. Now 1% CPI after heating would be yogurt-like structure. So it's still a strong gel, but it's even stronger when you have higher protein, because the protein is actually helping the gel formation by heat-induced protein denaturation. So that's the mechanism that we were proposing there, that the salt actually improve some of the solubility of the interfacial protein and then make them more repulsive, provided some steric stabilization. When you had vinegar, the pH was lower, they are positive charge, but although the protein become more hydrophobic, but they have higher electrostatic repulsion, so they can become liquid. When you have both salt and vinegar, then they are aggregated, and because of the acid-induced protein hydrophobicity and then salt-induced charge screening. And when you have heated emulsion, now the protein denature at the surface, and that form the gel, and the droplet now take part in the gelation. So that's our research on canola protein. So let's move our focus on meat emulsion. Now, meat emulsion are the main structural component in processed meat. So the particle size are reduced by mechanical means, and this type of meat system are, you commonly composed of lean meat and fat and other water, salt, spices, and other binders. So here is a schematic diagram of meat. So you have fat droplet with muscle fiber in the continuous phase forming a gel, and fat actually enhances the gel strength. So here is another microscopy taken in our lab. Now, so the question we ask is, if this is an emulsion, can we make then plant protein stabilized emulsion and use that as a create a meat emulsion type of structure? Also, can plant protein stabilized emulsion replace animal fat in meat matrix? So can we replace this animal fat droplet with plant protein stabilized droplet? So that can improve the nutritional benefit, reduce saturated fat, cholesterol. That was our question in this research. So this research was done by Dr. Fatima Kaivenar, and so she is a postdoctoral researcher in our lab. So what she did is she used faba bean protein, which contained about 58% protein. And then she made 48% canola oil in water emulsions. We use 4 different types of protein concentration, and the pH was kept as the original pH of the. And then we prepared emulsion, in this case using a polytron blender, because we didn't wanted to create a nano emulsion or very small droplet in this case. And then, similar to the other research, we added various amount of salt. We changed the pH. I'm going to report only one pH here just for the time. And then we heated the emulsions, and also we tested their fridge thaw stability. And so you can see here that the droplet size didn't really change with protein concentration. These data are protein suspension, so just the protein, not emulsion. So you saw that what we saw is the protein suspensions are large aggregate. They are also homogenized because we wanted to compare. But when you have oil, because now they absorbed on the oil droplet surface, their droplet size is significantly lower compared to the particle size of the protein suspension. And the charge. And this has reported before that protein, when they absorb on the droplet surface, they usually open their structure, they are more charged when they are on the droplet surface. Then we added salt. We can see that as soon as you add salt, different amount, the zeta potential decrease, salt-induced charge screening. And I'm showing you here data for 2 different emulsions, 6 and 10% protein stabilized emulsion. The droplet size for 6 and 10 didn't really change that much. And the protein suspension also had a higher aggregate size compared to the emulsion droplet size. So let's look at some of the gelation data. So here you can see the control emulsions they are rheologically gel, G prime higher than G double prime, but they are weak gel. You don't see much linear viscoelastic region. Adding salt, interestingly, actually decreased the gel strength. The emulsion become weaker with salt. But if you heat them, you can see that even the control without salt, from a weak gel, it becomes a strong gel due to this linear viscoelastic region. This is the characteristics of a strong emulsion gel. And so for all of these, as soon as we heat them with and without salt, their gel strength significantly increased. And 10% protein stabilized emulsions are also higher gel strength compared to the 6% protein stabilized emulsion. We looked at their microstructure. In unheated emulsion, not much change with addition of salt. With heated emulsion, we saw significant, droplet aggregation, protein aggregation, and some droplet were so packed that they deformed their structure. And that was the basis of, improving gel strength by inducing protein and droplet aggregation. But we had to make sure that, as I said, droplets don't fuse together, don't get coalescence. Then we changed the pH to an acidic pH to see whether that can induce gelation. So what we saw is that, acidic pH can induce the droplet size. So the aggregate size was higher compared to the 6.5. So again, acid-induced protein hydrophobicity, that could lead to this kind of more attraction between the droplet. And you can see that just by changing pH, you can significantly improve the gel strength. This is a strong gel, but this one not. And so gel strength increased by acidic pH. And again, by heating, similar to the other one, also heating always improved the gel strength. So now looking at the microstructure, so here you can clearly see that at pH 3, the droplets were more aggregated, but as soon as you heat, the proteins were extensive aggregation between protein and protein-coated droplet that gives you that strong gel-like structure in acidic emulsion. We then looked at fridge-thaw stability, and we found something really interesting, that these emulsions, okay, they are very good. They can form strong gel, but if we want to use them in meat, the meat will be frozen and they will be thawed. And we wanted to see whether our emulsion can survive that freeze-thaw process. And they were not. These were the regular emulsion without salt, with sodium chloride at the 3 different pH. And after the freeze-thaw cycle, we centrifuged the emulsion and saw extensive oil coming out of the free oil. So the emulsions were, almost completely destabilized. So because that, what happens is during freezing, there are a lot of ice crystals that push the droplets to as an unfrozen. The ice crystals can penetrate the droplet, can lead to droplet flocculation. So again, we need to have a strong interdroplet film that may be able to protect. So then what we did, that we heated all this emulsion, and our hypothesis was that heating this emulsion can give a strong interdroplet film, and that can prevent maybe freeze-thaw destabilization. So, and we actually saw that. So you can see that from a very high oil separation, we barely got any oil separation. With low pH, we had little more destabilization because droplets are more aggregated, but still, which is better than without heating. So that tells us that heat treatment prior to freeze thaw significantly improve emulsion stability, and the heat-induced denaturation led to increase in interfacial strength, and provided the structure against the fridge-induced emulsion destabilization. So then we realized that, okay, for our meat product, then we have to have these heated emulsion before they go into the meat processing. So, so the meat research where we replaced animal fat in bologna with structured emulsion was done with collaboration with Dr. Phyllis Shand of our department. Um, so what we did is, based on emulsion, gelation, and stability, the 10% fababin protein stabilized 40% canola oil emulsion was selected for bologna meat processing. But because it's a pilot plant processing, we cannot use a polytron blender, of course. So we used a bowl chopper, and we found out that they were as efficient as a polytron blender in getting the right droplet size. And then after we made the emulsion, they were packaged in casings and heat treated. And this is how they look like. So this is just the heat treated fababin emulsion. And as you can see, they are like a strong gel, like solid cubes. You can cut them in cube and mix with meat. And that's exactly what we did. So this was very exciting. And so what we did is in bologna processing, we either mixed with meat in the pork meat, lean meat, we either mixed with emulsion or pork back fat. And we use appropriate control to have a high fat and low fat animal fat versus emulsion versus canola oil or cooked emulsion. And then we did all the tests. I'm not going to go into too detail into meat analysis because of time. Some of the photos of our emulsion meat, bologna, you can see they looked good. We did some tests. It appeared that the emulsion in cooked emulsion, the one that we heated, was all very much similar to the low fat emulsion, because low fat and cooked emulsion have same amount of fat. We are still working on this. This is a research in progress, so I'm not going to talk more than this for today's lecture. So the last one I want to talk here today is research by Dr. G.M.Chi Doan, who was a postdoc in our lab, and she developed pea protein nanoparticles for improved emulsion stability. Now, before that, how are we doing with time, Dr. Nickerson?
It's 12:41 right now. So maybe 10 minutes?
Okay, yeah, that should be more than enough. Thank you. So what she did is she synthesized pea protein nanoparticles in using an ethanol desolvation process. And then she improved the redispersibility of the nanoparticle by again homogenizing the particle, and then used the PPN to develop highly stable Pickering oil in water emulsion. Now, Pickering emulsions are little different from the classical emulsion that we discussed at the beginning of this lecture. So classical emulsion, you have this emulsifier adsorbed on the oil droplet surface. We all know in Pickering emulsion, instead of traditional emulsifier, you have particles. So the particle can coat the oil droplet. Researchers have shown that there are various advantages of that, mostly that the interface is very strong because of the particle stabilized. So it can prevent coalescence and freeze thaw destabilization. So we wanted to see that whether, first of all, whether we can create nanoparticle from plant protein. Previous researchers have done that with, whey protein, with gene protein, but we wanted to see pulse protein, whether they can also do that. So what we did is we had a isolate solution. We centrifuge, we only took the soluble fraction, so it became 3% protein solution. And then we added ethanol at 1 ml per minute and with stirring. So what ethanol did is ethanol introduced, changed the polarity of the solvent, and the protein gets aggregated, and it leads to increased protein-protein aggregate interactions than protein-solvent interactions, and leads to formation of protein particle, which we centrifuge. We precipitate the particle, we took the particle and then dissolve them in deionized water, adjust pH from 3 to 9, homogenize them, and then we found that the pH 6, 7, 8, 9, the pea protein particles were highly stable dispersion, they have high charge, and then we use the pea protein nanoparticle dispersion at those 4 pH, and we made oil in water emulsion. So these are the PPN characterization. One of the challenge was that these nanoparticles, they were extensively aggregated. If you measure their particle size using laser diffraction particle size analyzer, impossible to get the right particle size. So we did some cryo-SEM work at Ghent University. Thanks to Professor Kuhn Dieting. And so we did some image analysis and measured the particle size, and we found that the surface average particle size is about 100 nanometer. We got this particle, but if you measure them separately, you will never get that particle size. Now, then we made emulsion. So simple, we made 5% oil in water emulsion with 28% ppn dispersion at 4 different pH. Just to briefly give you some data so this is the ppn emulsion. So you can clearly see that the droplets are covered with this particle, not a traditional looking emulsion. So this is the PPS which is the soluble pre-protein. So here you can see the droplets are much smooth surface, but in the PPN, they were coated with nanoparticle and they were very different compared to the PPS emulsion. We did some analysis looking at their droplet size. You can see that the PPS emulsions were a little bigger than PPN. They were, lower droplet size. Their zeta potential of the PPN nanoparticle stabilized emulsions were also higher compared to the soluble P protein emulsion. We also looked at their storage stability. So we found that initially the emulsions were obviously stable. They have similar droplet size. Now, after 1 month, you can see the PPS emulsion, they cream, their creaming separation, the PPN still stayed stable, the particle size, droplet size remained similar. Even after 3 months, you can see extensive creaming, phase separation, almost clear aqueous phase. But the PPN emulsion stays very similar. The droplet size didn't change. So it is a very stable emulsion compared to PPA, soluble pre-protein. So nanoparticle definitely improved the emulsion stability against creaming, and it provided much stable emulsion. Then something interesting, what she tried is she uh, did some time-lapse video of emulsion thawing after freezing. Because one of our hypotheses was that the pea protein nanoparticle will provide strong interface, which can actually prevent the freeze thaw destabilization. And you saw that. So this is a time-lapse video. I'm going to skip a little bit. So you can see right at that moment, the PPS emulsion, you see there is a phase separation happen, droplet coalescence, they are creaming, but the PPN emulsion remain, very stable. So this was really exciting that, the PPN emulsion remained stable. So this is what, we propose that the PPS emulsion, their droplets are not that strong, but the PPN emulsions, they were forming a strong interfacial layer, and the proteins were not, denatured. They were not removed from the droplet surface, and they were able to prevent ice crystal penetration, so provided stable emulsion. So I'm going to end here just to summarize very briefly. So to create structured emulsion, so various strategies can be utilized to induce interdroplet attractive interactions. You can add salt, you can add change pH, you can control droplet size, heat treatment. There are other things as well, but we just talked about these few in this presentation. But it is absolutely critical to prevent droplet coalescence. So we need to make sure the interfacial membrane stays very strong. And obviously, plant proteins are very much capable of to create the structured emulsion for various food application. So with that, I want to thank our lab who does all the work. I'm just presenting their work here. Fortunate to have such a greatly and motivated group. So this is the latest picture last year around this time, so before COVID, so no mask at that time. And also want to thank funding agency NSERC, Canada Foundation for Innovation, Government of Saskatchewan. Obviously, all these pulse protein research are funded by the Government of Saskatchewan, Ministry of Agriculture. Saskanola funding the meat research as well with Government of Saskatchewan and our University of Saskatchewan for their support. Also want to thank Pleasant Valley Oil Mills for their donation of canola, cold pressed canola meal, and AGT Food, as always, for their supply of endless supply of protein for our academic research. So thank you, everyone. And if you have any questions, I will be happy to answer.
Thank you, Dr. Ghosh. We'll now have maybe 10 minutes for questions, so feel free to unmute or write it in the chat, and maybe I'll get the process rolling. Supratim, have you done any work looking at the digestibility of your plant-stabilized protein nanoemulsions or gels?
We have so far done on nanoemulsions, and we published one article on that, two articles actually. So yeah, so they were interestingly, the digestibility was little lower than the lipid digestibility, little lower than dairy protein stabilized emulsion, if I can remember correctly. Yeah.
Okay.
But yeah, that's an interesting suggestion. Yeah, we should look more into that.
Perfect. I'm just about to read something from the chat. I can feel someone typing out something now. This kind of feels like our lectures that we give. No one asks any questions.
I know.
Okay. I guess if there's no questions, thank you, Dr. Ghosh, for a really nice talk. I think it was very informative for the group. We had a good showing of attendees, up to 71, so that was really great. I should mention that our next seminar series is April 27th. Carla Prado from the University of Alberta will be coming to talk about the role of protein intake in optimizing health outcomes. So thank you, everyone. And again, thank you, Dr. Ghosh, for a great talk. And yeah, we'll see you in April. Thank you very much.
Thank you, everyone.